Hello, this video is about carbohydrates, their structure, role, and characteristic reactions. Let's start from the outline. We will take a look at classification, so we'll take out groups of carbohydrates. We'll look what isomerism means in respect to that group. What are the examples of isomers and what are the biological consequences of isomerisms in carbohydrates? We will take a look at hydrolysis and condensation reactions and examples in living organisms. And then finally, we will explain the role of carbohydrates in living organisms. Let's start from the question, what does it mean, carbohydrates? Carbohydrates, as the name suggests, that there is a carbon, so carbo, okay, so there's a carbon, and hydrates. So basically, it means that each carbon in a molecule of carbohydrates is this specific relation to hydrogen oxygen. So we may say they are hydrated carbons, which is actually, of course, very large simplification. However, the general formula of carbohydrates is a CxH2OY. You may find this formula in a textbook for chemistry. Okay, let's take a look at the groups. The first most simple group of carbohydrates are mono and disaccharides. And in case of monosaccharides, in important and living organisms, we distinguish the three groups. From monosaccharides, we distinguish thioses, so carbohydrates made of 3 carbons. As you can see, they do not form a ring, but basically they are linear. Carbohydrates made of 5 carbons, so 1, 2, 3, 4, and 5, forming a ring, and hexoses, so compounds made of 6 carbons in a chain, also with a ring. What are the examples of the molecules in living organisms? In case of thioses, one of the most famous trios is a phosphoglyceraldehyde. This is the first stable product in photosynthesis. We will meet this molecule when we will talk about cellular respiration and photosynthesis, especially photosynthesis. It's a very important component of the Kelvin-Benson cycle. Pentoses, the most two famous components of nucleic acids, is a ribose and deoxyribose. And from hexoses, in living organisms, we find glucose, fructose, and galactose. Of course, this is just an example of carbohydrates, very simplified, presented to you mainly because of IB requirements in molecular biology. Okay, what does it mean? What does it mean isomer? In case of an example of glucose, the isomer of glucose is made of exactly the same number of atoms of carbon, hydrogen, and oxygen, but with different spatial arrangements. In case of glucose, we distinguish alpha glucose, which has a hydroxyl group at carbon number 1 directed in opposite direction to the 6th carbon, The consequences of this isomerism in case of glucose is that if these hydroxyl groups are directed in the same on the same side of the ring, they will form different shape of bonds and different shape of structure of entire molecule than in case of alpha beta isomer. And that's why the cellulose, which is a polymer of beta glucoses, looks differently than starch or glycogen, and cannot be recognized by salivary amylase or any amylase enzyme. So that's why cellulose is not digested by this enzyme. Very frequently in biology, some isomers of organic compound has a different role than the other one. One of the most famous examples is the vitamin C, which is a L isomer of ascorbic acid. Its D isomer is completely biologically inactive molecule. How we can test for the presence of reducing sugars? Reducing sugars include glucose and fructose, the sugars that are able to reduce substance. The reaction is called the Benedict test and is based on a reaction of oxidation of copper to sulfate into copper 1 oxide with a reaction in which the color is changed from blue through green and orange to the brick red, where the copper is reduced. There are even some ability to estimate the percentage concentration of reducing sugars. Here you can see the reference colors. Of course, this reaction with colors is very subjective and has a limited value in case of a qualitative you can assess the more or less the concentration, but quantitative results are not very reliable. Okay, let's take a look at the role of monosaccharides. The phosphoglycerolaldehyde, so the first product of photosynthesis in plant metabolism, is used to combine into glucose. And of course, we know that glucose has a very wide role in living organisms. It will be mentioned a little bit later. From pentoses, ribose is a component of RNA nucleotides, so ribonucleic acid nucleotides, so builds RNA, such as ribosomal RNA, messenger RNA, or transfer RNA, and is also found in ATP, so this universal currency of living organisms. Here you can see the structure of a ribose, very specific arrangement of hydroxyl group at carbon number 2 and carbon number 3. Deoxyribose is very similar to ribose. However, the only difference is that it lacks hydroxyl group at carbon number 2. There is only hydrogen here. If you wonder how the nucleotide looks like, here you can see the nucleotide, so basic units of a nucleic acid with a phosphate, pentose, sugar, and nitrogenous base. However, nucleic acids will be described in next one of the next topics. Let's take a look at glucose. Of course, we all know that this is direct energy source in cellular respiration. However, it also is a building block for more complex compounds, such as disaccharides, that will be mentioned in a minute, and polysaccharides, such as starch, glycogen, and cellulose. Glucose in general are so carbohydrates, and most of them, actually, at least, because cellulose has a structural role. Their role is to provide energy for a cell, energy for an organism. They may even store this energy for some time, but they are also known as free calories, because our organism basically may synthesize carbohydrates. However, it uses them mainly to produce energy. Yeah, that's why you can cut the carbohydrates in your diet and still be able to function quite effectively, as our organism can synthesize glucose from other nutrients. The fructose formula looks like that. So you can see that it's slightly different because there is a one carbon here outside the chain. The ring is similar to this in ribose. However, we still see that there's one carbon here, second here, third, fourth, and fifth here, and here's the sixth one. Fructose is a fruit sugar, so found in fruits such as grapes or pears or apples, and it's also a component of sucrose, so this table sugar we use in our kitchen. Galactose is a component of lactose, which is sugar found in milk. I think you know the lactose from the issue of a lactose intolerance. This will be in our future lessons, and it has also some structural role in cells, because it is present in carbohydrates found in glycolipids and glycoproteins. We remember from cell and membrane topics that it is the components of membrane. Glycolipids and glycoproteins are engaged in cell-to-cell communication, and for instance, the blood groups in human. Okay, let's take a look how monosaccharides join together to form disaccharides. Here you can see the reaction in which two glucoses, by condensation, produce maltose, which is a disaccharide, in which there is a glycosidic bonds. This is characteristic bonds in carbohydrates. Okay, you can see it in here. All right. And water is produced as a byproduct. That's why we can have a on daily basis connotation when condensed milk. Yeah, condensed milk contains less water than the common milk. Okay, in case of this link, because it is between carbon number 1 and carbon number 4, it's 1, 4, and this is alpha glucose, therefore it is 1, 4 alpha glycosidic bonds. This bond will be detected, will be recognized by our amylase enzyme in our digestive system, so we digest maltose effectively. And we digest starch, because you will later see that maltose is a main component of the starch. Other disaccharides are formed when glucose is bounded to fructose. We obtain a sucrose. Yeah, the sugar found in, for instance, sugar beet or cane. Yeah, and when glucose binds to galactose, lactose is produced. We heard about lactose a minute ago. The reverse reaction, so breakdown of disaccharide into monosaccharide, is called hydrolysis. So water is used to fill the gap between the carbon here and oxygen. We know from the previous lessons that hydrolysis is an example of a catabolic reaction, because more complex compounds is broken to more simpler, whereas the condensation is an example, maybe use an example of a anabolic reaction. Okay, this table shows you more details in case of a structure of disaccharides. So what exactly monosaccharides are used to build them, and what is their role in different organisms. I would like to move to polysaccharides now. So more complex carbohydrates, from which the first one is starch. Starch is famous from being the presence in plant cells, especially in some organs of plants, such as in potato tubers. Yeah, for instance, you can see here it's a potato drawing. Starch has two main subsections. One is called amylose and the other one is called amylopectin. You can see the shape here. Amylopectin is more branched, and amylose is just a chain of alpha glucoses. The straight chain is because between glucoses there are only alpha 1 4 glycosidic bonds, whereas the branching points in amylopectin are because from time to time, yeah, here in some of these places, there is not all, there is a 1.6 glycosidic bonds. It makes the chain branched. Starch, because it's a very large molecule, because contains hundreds and thousands of glucoses, is very slightly soluble in water, so it cannot form a solution. This is very important for plants because they can store starch in unlimited amounts, because starch, being insoluble, does not affect osmolarity of cells. Equivalent of starch in animals is glycogen. It's quite similar to starch because it is also made of alpha glucose linked by 1.4 glycosidic bonds, but also by 1.6 glycosidic bonds. So you can see that this molecule has also branched chains. However, it is relatively smaller than starch, so its solubility is relatively higher than starch. The third polysaccharide in our lesson is the cellulose. We said that it is made of beta glucoses linked by 1.4 glycosidic bonds. This substance is not digested by amylase. The only enzyme that digests this substance in living organisms is called cellulase, and it's produced by microorganisms, protists, and bacteria found, for instance, in cow's stomach or in termites' digestive system. Because of its difference in monomer, because of beta glucose, the shape of a link is different, and in reality, it forms very straight fibers of beta glucoses. You can see them here. Because of its regularity, the hydrogen bonds form between adjacent chains, and these bonds strengthen this made of many cellulose fibers, cellulose macrofiber, fibril, made of single cellulose strains. That's why cellulose has a very important role in cell wall of plants. By this table, you can see the comparison of the three polysaccharides. Again, we can see the similarities, and we can see differences. As all questions compare and contrast in our program, we needed to show both of them, the features that link the molecules and features that made the molecules different. That's all for today. That's all for carbohydrates, actually. The next lesson will be about the lipids. Thank you and see you later.